Sidekicks aren't here today. Lowering, yeah. Lightweights. Okay, let's get started. So we are about a week and a half away for your next exam. So it's not this Friday, it's the Friday after. So still kind of getting closer on the horizon anyway. That next exam is the same day as the drop date. So drop date Friday, October 28th. So unfortunately, we won't have that data to know, to give you more information whether to make that decision, whether you should drop or not. But something to think about whether you should drop or not. If you are failing and don't see a likely way that you are not going to fail, then it's probably a good idea to drop. Okay, so basically what this means is after October 28th, if you decide you want to withdraw, that is very difficult to happen unless some emergency has come up since the drop date. Okay, so the university doesn't like you to do that. I think it looks bad for the university as a whole in terms of, I don't really understand the reasons behind it, but they definitely, sometimes you can get away with it, but they don't like to let you drop after that date. So basically your grade will show up on your transcript. Question about that? Yeah, so to get a C, I believe a C minus is 70, 71 or 70. So essentially, if you get a 71.5, that will round you up to a 72, and that's a C for me. So yeah, I mean, if you need help running the numbers, if you want some advice on what you think you should do, we have a week and a half before that. Of course, next Monday and Tuesday, you won't be around. We won't have class because of fall break. So kind of start thinking that way. It has to be in the paperwork signed by 5 o'clock, and that's a pretty hard deadline. So yeah, just wanted to bring that up. Not trying to tell you that you should or shouldn't, just it's something to consider for some of you. All right, and I thought we would do a couple of practice questions. I know for that chapter on microbial control, as well as our antibiotic stuff that we're getting into, sometimes it's just kind of an information dump, and students ask me, I don't know how to study this, I don't know what level. I'll try to give you a few practice questions, but here's a good example. This was on last year's test, and I thought, well, I can't use quite as many questions as I did last year, so I'll show you this one. Vaccines, heat sensitive. So how could we sterilize a vaccine? Hopefully you've been staring at this maybe for a couple minutes, but of course it says it's heat sensitive. So that means you can't use anything that requires heat. So pasteurization, autoclaving, those are both heat methods. Freezing is not going to sterilize. Okay, that really just stops microbes from growing, doesn't get rid of them. So you're down to either lyophilization or filtering. Lyophilization, remember, is when we freeze something really quickly and then put it in a vacuum to draw off the vapor. So it's kind of like another way to say freeze drying. It just eliminates that liquid step so that those water crystals don't form. So again, you are dehydrating. Usually when we dehydrate something, we don't still call it sterile. We might dehydrate food to make it keep for longer, but it's not necessarily sterile. So that's not going to be your best option. I showed a picture of a syringe that had a filter in it. It's like if you needed to sterilize some drug, you put those pores really small in that membrane that you're forcing the liquid through. So again, I like questions that have to make you kind of think through a lot of different things and kind of synthesize that information. Sometimes there's some confusion on that therapeutic index that we talked about last time. Those are kind of intro to antibiotics. Say drug A has a therapeutic index of 28 and drug B has a therapeutic index of 104. So which one is considered a safer number? Remember, we figured out the therapeutic index as the dose that's toxic divided by the dose that the drug works. We called it the therapeutic dose. Okay, that toxic dose is always going to be larger than the dose that works. So in effect, you know, the higher the therapeutic index, the safer it is. Okay, so yeah, I said that right? So you're going to be you want to use drug B because that's the safer one. That's what it comes down to. Okay. All right, so we'll pick up where we left off last time. We had just started talking about Beta-Lactium drugs and how those work. We went pretty in depth on the mechanism of action on this one, just because it's a huge class of drugs that are commonly used. They actually bind to the enzyme transpeptidases, which remember are necessary to bind back those amino acid bridges that are reformed during the growth of the cell. When the new peptidoglycan subunits get inserted, those bridges have to be disrupted and then kind of built again. So the beta-lactams again bind to that enzyme transpeptidase. Otherwise, we get the cell wall being kind of fragile, and then it's susceptible to some osmotic forces. If you look at that on a little bit more molecular level, that might help you understand exactly what's going on here. So here's, usually we didn't really talk about which amino acids are in those peptide bridges because there's some variation there, but usually there's two amino acids, alanine, alanine. Amino acids have like a right-hand form and a left-hand isomer. This D-ALA is the isomer that's not commonly found in proteins. It's one of the only places it's found is in bacterial cell walls. So that's not very common in nature. And that is the normal substrate for the enzyme transpeptidase. I guess I'm switching to purple today. Big change in my writing. Okay, here's the structure of penicillin, and if you superimpose the penicillin over the D-ALA, you'll notice here it's got the exact same structure. Okay, so you could lay those right on top of each other. So actually, penicillin, what it does, it binds to the active sites of the enzyme transpeptidase. Okay, so essentially what it is, it's kind of like being a competitive inhibitor. We don't actually call it that because it's permanent. We'll say, but permanent. If it was just binding for a time and then it let loose, and we called it a competitive inhibitor, but it binds to that active site of transpeptidase and it doesn't come out again. So it's permanent. So in other words, I tried to kind of write that down here. Penicillin has the same structure as that D-ALA, so binds to the active site Okay, it's only working on growing cells. If that peptidoglycan cell wall is not growing and expanding, okay, bonds have to be cut for it to do that, then penicillin isn't going to have any effect on the cell at all. And again, it's not really the penicillin that lyses the cell, it makes the cell susceptible to osmotic forces there. So it's going to work best against cells in log phase, cells that are actively growing. Okay, so we're going to start our little kind of mini pharmaceutical course here, where we're going to go through about 14 or 15 different antibiotics and think about especially how they work. Okay, so if you know one thing about those antibiotics, you're gonna want to know the mechanism of action. So there's only about 5 or 6, depending how you count it, mechanisms of action. You're gonna want to focus also on the advantages of using the drug. I'm going to give you some other information. I talk about the spectrum of activity, or whether it's acid labile or not. I just want to get you comfortable with thinking about those things. I don't need you to memorize for each drug what the spectrum of activity or whether it degrades in the stomach acid or not. I just want you to understand what those terms mean. So think about how we use the drug. If there's one infection, oh yeah, we always use this drug for this infection, like that's something to know. Or if there's some big advantages to using the drug. Also, if there's an interesting side effect or a reason that we don't use the drug, then that's something to kind of focus on as well. Okay, so let's start with our narrow spectrum penicillins. So we're going to start with the very first antibiotic that was developed. We call that penicillin G. What does the G stand for? It doesn't really stand for anything. Apparently, when they were first developing penicillin, they had sort of different variations of it, maybe variations in how they extracted it from the penicillium mold. The A, B, C, D, the G one worked best. So that's the one that they used. Structure over here, don't memorize structures. I don't have that memorized, but what you should notice, remember we talked about, oh look, it has a beta-lactam ring. So I can't tell you, what kind of penicillin this is, but I know that it has a beta-lactam ring, so it's beta-lactam. The first penicillin that was developed, it was only effective against gram-positives. So that's why we consider it a little bit more narrow spectrum. Okay, only against certain organisms. Flory and Chain, the ones that figured out how to mass produce penicillin, argued about whether they were going to patent it or not, and they ended up not. So that would be unheard of today, but they didn't. So right away, it was very cheap. It was accessible to anybody that needed it, basically, very quickly. It reaches concentration pretty quickly in the cells. It penetrates well, but it actually, I'm going to skip down here, it requires injection because it's acid labile. So remember, that means it's destroyed by our stomach acid. So we can't take it orally. And anyway, even if you could, it doesn't get absorbed well through the small intestine. So you have to give massive amounts of the drug for it to reach the concentration well if you're going to give it orally. So that didn't work out. I can remember my dad, who was born in the 40s, so he's probably about the age of your grandparents. Next time you see them, ask them if they remember getting injections of penicillin. I can remember my dad thinking about Doc Tucker coming to the house. It's another thing that doesn't happen ever anymore. But the doctor came to his house when he was ill, and he had to get shot of penicillin in his rear end. So kids now have it pretty good. They don't have to go through that. But that was again early penicillin. big disadvantage now. It's not used very commonly because resistance is so widespread. And even Alexander Fleming predicted that that would happen right from the get-go. So he was right. We're going to talk about this, why there are bacteria that have gained the ability to make a certain enzyme called beta-lactamase, and we'll see what that does in a minute. The kind of, if you've ever taken penicillin, the kind that you would have taken is penicillin V. When they grow the mold up on a little different substrates, it produces a slightly different product. And so whether you call that semi-synthetic, people kind of argue about that. But it is just slightly different. I think there's just one little side change difference, and that actually can be taken orally. So yeah, question.
When a bacteria gets like resistance to penicillin, does it get resistant to all types or switch the types?
So good question. If a bacteria gets resistant to penicillin, is it resistant to everything? Thankfully, no. that can happen, but typically when an organism gets resistant to penicillin, it's because they can make a certain enzyme. So I'll talk about that here. There's different ways that we are going to talk about how bacteria gain the ability to be resistant. But one pretty common way is they acquire the ability to make this enzyme beta-lactamase, which sometimes is called penicillinase. I usually call it beta-lactamase. So here's the drug penicillin, and if the organism makes beta-lactamase, then it cleaves this bond right here in that beta-lactam ring and opens it up. Okay, and that's enough to change the structure of penicillin so it no longer will bind to the active site of transpeptidase. Okay, so beta-lactamase is, again, the most confusing thing when we talk about resistance is who is doing what. Beta-lactamase, that's produced by the bacteria. How do they learn how to make that? Usually they get instructions for that protein from a plasmid that's been shared horizontally from another cell. Okay, and then again, that's enough to make destroy the activity of penicillin. Okay, but there are ways that bacteria can become multi-drug resistant, and stay tuned, we'll talk about that next time. Okay. And then one of the early derivatives that we had of penicillin that we don't use anymore, but we came out with methicillin in the early 1950s. We don't use this anymore just because it's basically everything's resistant to it now. But I like to mention it because there is a strain of Staphylococcus aureus called Mrsa that probably most everybody has heard of. It stands for methicillin resistant Staph aureus. That first showed up about 1961, okay, when we still were using methicillin. So that's how it got its name. Oh, here's a strain of Staph aureus that now, you know, is not inhibited at all by methicillin. So who cares if it's resistant to methicillin now? That's not a big deal because we don't use it anymore. But the point is, it's resistant to lots of other commonly used antibiotics, especially the beta-lactam. So it's a huge problem. Mostly skin infections, but can result as after surgery, things like that. Okay, but originally it had a little bulkier side chain, I think, that kind of blocked the action of the early beta-lactamases. Some side effects, it had a lot of kidney toxicity associated with it. Acid labile, so had to be injected, either by syringe or through IV. So again, really not used much anymore, but that's where the Mrsa name came from. So we need to stop and talk a little bit about penicillin allergies. they're fairly common. When you look up how many people are allergic to penicillin, you get very different numbers. I've seen as high as 10% of us. We probably think it's more like around 1 to 2% of us. A lot of people have been told they're allergic to penicillin after they got a rash, after they used it. And that rash could have been due to something else, but it could have been due to penicillin. And maybe the next time that person has penicillin, it could be a much more severe reaction. So usually any physician won't prescribe any beta-lactam antibiotic after that allergy has been, kind of said that a person has it. More and more, it's at least for kids, if they suspect kids have a penicillin allergy, you can go to an allergist and have them go through a whole battery of tests that starts with the pricking on the back. And then if that's not showing a reaction to penicillin, they'll inject it intradermally. And if that doesn't show a reaction, they'll give it orally, like 3 different doses of it. And if the kid passes that, then okay, nope, they're definitely not allergic. So that's something that they're a little bit more willing to do now. If you get a little rash, well, maybe that's not that big a deal. Hives are a little bit more of a deal. Some of you know Dr. Baxter. He teaches this class also. When he was young, I don't know, a teenager or a kid, he had hives for 9 months after he had penicillin. So he should not have penicillin ever again, for sure. Sometimes there's other things like fever or even diarrhea. And it's kind of hard. Well, was that from the penicillin or was that from the infection that you're trying to keep to treat? There's a small percentage of people that actually will make antibodies to the metabolized product of penicillin. And that can actually cause a life-threatening, anaphylactic shock. When a patient goes into shock, their blood pressure goes way down and basically their organs shut down. About 200 Americans die every year from an allergy to some kind of penicillin or beta-lactam. Usually if you're allergic to penicillin, you're allergic to all the beta-lactam drugs. So it's something, people are different. People are allergic to just about anything you can think of, but that's one that certainly has the potential to be very serious. There's probably a lot of us that think we're allergic that maybe we're really not. Okay. And then thinking about broad spectrum penicillins. So again, these are kind of the later generations, we would say, of penicillin. Amoxicillin. This is really, really commonly used today, especially when kids have an ear infection or strep throat. Again, very, very similar to the structure of penicillin. Note that beta-lactam ring in there. It's acid stable, so we can give this one orally. It's the one that's they make it bubblegum flavor. I mean, I can remember taking penicillin as a kid, and it tasted awful, like awful. Like mom would give me a saltine cracker to try to get the taste out of my mouth. So again, you all have had it good growing up. You didn't have to get shots. Your drugs tasted good. It absorbs better than the original penicillin. And I think you still typically have to dose this maybe 3 times a day, but maybe it's not as often as some other drugs. I think we've been using amoxicillin since about 1972. So we've used this for quite a while. Resistance is becoming a problem. So oftentimes amoxicillin is actually packaged along with another drug, clavulanic acid, which is actually a beta-lactamase inhibitor. Okay, and so when you package the amoxicillin together with clavulanic acid, The name brand of that product is Augmentin. Okay, if you augment something, that means you add to it to make it better. Okay, so we've added clavulanic acid along with amoxicillin in an effort to lengthen the life of amoxicillin so that we can keep using it and not everything becomes resistant. Question?
Beta-Lactamase is the enzyme that makes beta-lactamase.
Yes. So beta-lactamase is the enzymes that bacteria make in order to destroy the activity of the antibiotic. Yep, the beta-lactam antibiotic. Good, another question. It's a good question. Is there an average amount of time that it takes for resistance to build up? I don't know that that's like a linear thing. I know there's a lot of things that we do now to try to slow down resistance. And we will talk about that more on Friday, but there's a lot of infections that we might give a couple of different drugs that work a little different way, so that even if it becomes resistant to one way, the bug is still killed by the other drug. I think we are much more aware of it now, so maybe we're getting a little bit longer lifespan out of our antibiotics than what we originally did. But yeah, I don't really know the answer to that question, if there is an average amount of time. So good question. Okay. And then other beta-lactams, cephalosporins are a group of drugs that have that are used a ton. They've been around for a while. All of the cephalosporin drugs, they begin with cephala prefix, so that's how you know that it's a cephalosporin. And one example is Cephalexin. The confusing thing with a lot of drugs, they have like their name and then the company that makes it puts on a trade name. So for instance, Cephalexin is often known as Keflex. That may be a little bit more recognizable. These, like I said, they're beta-lactam, so they work the same way. They bind to the transpeptidases that leads to cell wall lysis. With this, you can see there's actually two variable groups here. There's an R group here and this X group over here. So there's two different spots where we can kind of mess with the structure there and make some semi-synthetic antibiotics that will give each rendition just a little bit different properties. So our early generation cephalosporins were very narrow spectrum. Like I said, right here, we have the early ones only worked on gram-positives. As we've kind of monkeyed with them and get second generation, third, all the way up to fifth generation, they've become more broad spectrum, but also have a little bit more toxicity issues. So kind of the later generation ones, if they have more toxicity, then we're not going to give them orally anymore. We're going to put them in the IV so we can dial in that concentration just right. The higher generations have it's not the not common ability to cross that blood-brain barrier. So not very many pathogens can do that, but the ones that do, we have to have a drug that follows it there. So used to treat infections like meningitis. Again, the later generations. Again, I think the earlier generations could be taken orally, but the later ones, because they have more toxicity issues, are typically injected. And that fifth generation, even though it's a beta-lactam, it's different enough apparently with those two side chains that it's effective against Mrsa. So that's one of our go-tos, again, late generation cephalosporins for Mrsa. And still a couple more cell wall inhibitors. I know there's our biggest, the most number of drugs here that we're talking about are cell wall inhibitors. Because remember, we don't have cell walls, so good selective toxicity. There's a couple of cell wall inhibitors. They're not beta-lactams, so they work in a slightly different way. but they still work on the cell wall. So vancomycin, again, really complicated structure. There's no beta-lactam ring there. It binds to those peptide bridges of peptidoglycan and just kind of is a physical block so that the transpeptidases can't come in there and complete those bridges. So again, more of a physical block there. Vancomycin only works on gram positives. So it's another drug that will typically work on Mrsa. We usually don't use vancomycin on our run-of-the-mill gram-positive infections like strep throat. When it first came out, it had a lot of toxicity issues. We've gotten better at kind of purifying that drug, so we don't have as many issues, but we still kind of save this as, hey, when this other stuff doesn't work, our amoxicillin or whatever, and nothing else is working, then we'll try vancomycin. So that's in an effort to try to keep, lots of drugs become resistant to vancomycin. Because it doesn't get absorbed very well through the intestine, we usually are going to administer it by IV. The one exception to that is that this is often the drug that's used to treat C. diff. Okay, and because that's an infection in our intestine, our large intestine, we have to give it orally if we're treating C. diff. Anytime you have a strain of organisms that's found to be resistant to vancomycin, that's considered a medical emergency. So VRE, that stands for vancomycin resistant enterococci. Okay, again, this is probably an infection that would be acquired in a hospital. And again, that's a medical emergency because they don't have much left to try to treat that. Another cell wall inhibitor, bacitracin. When those new NAG and NAM sugar subunits, they're made inside of the cell, and then they actually are transported through the cell membrane through a very particular channel. Bacitracin actually acts to prevent those new sugar subunits of the growing peptidoglycan, prevents their transfer through that channel. So that's how it works. Again, this is narrow spectrum, only targeting gram positives. This drug in particular is quite toxic to the kidneys. So this is one that we don't ever swallow orally. So it's only used topically. And it's one of the antibiotics in the triple antibiotic cream. Okay, so anytime you get a cut in your skin, it's a good idea to put on some triple antibiotic cream to keep an infection from growing. Okay, so we actually have 3 antibiotics in there. That's not too hard to work out. One that's a cell wall inhibitor, and the other 2 antibiotics work by different mechanisms. So we have like 3 different mechanisms, and we'll talk about what those antibiotics are when we get to them. Okay, so I think, one more cell wall inhibitor, isoniazid. Kind of fun to say. This is a drug that's synthetic. So this is not biosynthesized. We make this in the lab. The way it works is that it prevents synthesis of mycolic acid. So I think your textbook actually categorizes this drug as its mechanism of action is that it prevents a biochemical pathway that we don't have. I grouped it here like our previous textbook just because mycolic acid is an important component of some bacterial cell walls. Remember, only bacteria, what kind of bacteria have mycolic acid in their cell walls? Yes, okay, narrow spectrum. It's only working on acid-fast bacteria. And so that's that genera called Mycobacterium. Okay, so we use isoniazid to treat infections like TB and leprosy. It has some side effects, nausea and headaches. which make it kind of hard to keep taking. Okay. All right, so that's our cell wall inhibitors. I found it useful to draw up a what do you call this? A concept map. That's what you call it. And so starting, here's our antibiotics, and then I've got the squares are our different mechanisms. In circles here are an example of a kind of a big class of drugs, and then the examples of the drugs that we have talked about here are out on the branches. So, and then so you're going to want to fill in here as we go the other things that you'll see at the end of those branches. For me, when I was first teaching this, it helped me to just draw up this concept map a bunch of times. You know, flashcards may be of some use as well, but just to learn those mechanisms. Okay, so we're done with our cell wall inhibitors now. Let's move on to another big class, protein synthesis inhibitors. It's probably the second largest class. Got a number of drugs that are protein synthesis inhibitors. In general, most of these are more broad spectrum. Okay, so the drugs that target the cell wall usually having something to do with the peptidoglycan, and most of them work better on gram positives because gram positives have more peptidoglycan. Okay, so these are a little broader spectrum. We talked about the kinds of organisms that produce antibiotics. Remember Streptomyces, that was that genera of bacteria that kind of act like fungi. That's one that produces a lot of the drugs in this class, or that genus of bacteria. Okay, so a big class of protein synthesis inhibitor are the aminoglycoside antibiotics. They bind to the small subunits of the ribosome. And so that's how, in different places, but that's how they inhibit protein synthesis. Like I said, they're fairly broad spectrum. They don't absorb very well, and they're acid labile, so they have to be injected. High toxicity. So what kind of toxicity? I found this little graphic here. If you look at it closely enough, you can kind of see what it is, but it's the ears boxing with the kidneys. Okay, so nephrotoxicity, that means toxic activity against kidneys, and ototoxicity, toxic effects to the ears. And on the boxing gloves are the different names of some of the aminoglycosides, like gentamicin and kanamycin. All the aminoglycosides end in that mycin suffix. That's how you know they're an aminoglycoside. Okay, so like I said, kanamycin, gentamicin. Well, they spell this one differently because it doesn't come from Streptomyces. It comes from a different bacteria. FYI, if that's interesting or not, I don't know. Streptomycin, we're going to talk about that one a little bit more closely here. And neomycin, I'll just mention this because that's the second drug in triple antibiotic cream. Okay, so again, we have bacitracin, the cell wall inhibitor, neomycin, and aminoglycoside protein synthesis inhibitor. Okay, we'll get to the third one. Okay, so looking a little bit more closely at streptomycin, this was one of the very early drugs that was produced. I think it was in the 1940s, like the late 1940s. Salman Waxman was the guy's name. He was a soil biologist. And he developed over 20 different antibiotics, or somebody in his lab, and he kind of got credit for it. So the story goes. But streptomycin was the first broad spectrum antibiotic. So this was the first antibiotic that had some activity against some gram-negative things, like the plague that's caused by a gram-negative organism. It's actually the first drug that had any effect on tuberculosis. So this was a real miracle drug when it came out. We thought we were going to eradicate tuberculosis. Didn't work out that way. But if you look at the structure here, complicated, but you remember the name, an aminoglycoside. It's an amino modified glycosidic sugar. Okay, so this is another way to draw sugar structures and that kind of collapsed ring. And again, we've got these amino groups. So aminoglycoside, amino modified glycosidic sugars. So it's kind of nice when the structure kind of makes sense for what we call it. It binds to the small subunit, so you can kind of see this down here, changes the shape of that subunit, and then it doesn't read the messenger RNA correctly, and the wrong amino acids get translated into that protein, that new polypeptide. Broad spectrum toxicity to ears. That seems like a strange side effect. If you're in anatomy, it actually works on the vestibular portion of cranial nerve number 8, I believe. Okay, so again, if you're in anatomy, that might mean something to you. So what effects does that cause? It may just be kind of a mild tinnitus, ringing in the ears when a patient's on streptomycin. It could cause some vertigo, which causes you to be really, really nauseous. It can cause hearing loss that's kind of transient, or it can cause permanent hearing loss. So we have to be pretty careful how we dose this when we use it. But again, this was a miracle drug, first thing that was effective against tuberculosis. First thing that worked against bubonic plague. We were making a lot of strides in getting rid of bubonic plague because that's carried by the fleas that live on rats. Okay, if you don't have garbage laying around everywhere, then you don't have the rats and you don't have the fleas. So increased hygiene had helped us there. Also, the first drug that was effective against tularemia, which it's a gram-negative infection that's passed by the fleas on rabbits. So might not have heard of that one before, but that is a potentially life-threatening infection as well. So again, game changer. We don't use streptomycin anymore for tuberculosis, and they may try it if nothing else is working, but there's just a lot of resistance to streptomycin now for that Mycobacterium tuberculosis. Okay, other protein synthesis inhibitors, the tetracyclines. There's a whole class of drugs called tetracyclines, like doxycycline. Anything that ends in cycline is going to be a tetracycline. So these are not aminoglycosides. There are other protein synthesis inhibitors. If you look at the structure here, again, don't memorize structures, but if I showed you that structure, 4 rings, tetracycline, that makes sense, right? So there's something that you can recognize there. Again, it targets, like aminoglycosides, they target the small subunit of that prokaryotic ribosome. I'll come back to this in a minute, but it actually binds right here at the docking site of the new tRNAs, that A site, and keeps new charged tRNAs from docking there. So essentially, a tetracycline is just going to keep a bacterial cell from being able to make new proteins. It won't kill it right away, but it will stop it from growing. And oftentimes that's enough to give our immune system a chance to tackle it. So, okay, again, broad spectrum, really cheap. That's probably one of the reasons that it's been so overused in agriculture. And we're starting to understand that maybe that's not a good idea. But when we give it to, say, chickens in their feed, it makes them grow faster. So not only keeps them from getting sick, it makes them grow faster, probably because it cuts down on some of the bacteria in their colon that basically they have to feed that bacteria. You don't have to feed the bacteria anymore. The animal grows a little bit quicker. So, but we're starting to understand that maybe we're overusing this drug. Most of the strains of Salmonella now are, you know, we would like to get rid of in chickens are resistant to tetracycline. So we're worried that this is one way that it's kind of, you know, trickles over into humans and gives bacteria resistance. So, but it's nice, you can give it orally. Side effect here, you can't ignore this kind of nasty picture. It affects developing teeth and bones. Okay, discolors teeth, makes, I think it kind of affects bones and a little bit, doesn't make them quite as strong. Okay, so for that reason, not given to children. Okay, we'll try something else. And in adults, when they take a tetracycline drug, it has phototoxicity. So in other words, adults are going to sunburn easily. Okay, so one of the common uses of a tetracycline is for acne. If you're on a tetracycline drug for your acne, they'll tell you whenever you're out in sunlight, you need to wear sunscreen, because again, you'll get sunburned a lot more easily. Doxycycline, it's a kind of tetracycline that's commonly used to treat gonorrhea right now, as well as one of the drugs to treat pneumonia. So I'm sure there's lots of other uses, but those are just some of the most common ones. Okay, and then our last group of protein synthesis inhibitors, the macrolides. Okay, this is the one group that will target the large subunit of the ribosomes. And again, we didn't talk about glycosamides here, but you know, it binds there and somehow keeps the messenger RNA from feeding through that ribosomal complex correctly. The structure here is complicated. We call this a macrolide ring. Okay, not as recognizable maybe as some other things. This is typically an example of a macrolide. There's not a lot of them, but the example that you've probably heard of is azithromycin. Sometimes we call it a Z-Pak. This is one of the most commonly prescribed antibiotics for you know, when you go to the doctor's office and you have like an ear infection or whatever, strep throat, the one that they would send home with you, azithromycin. It's nice to take because it has a long half-life and it remains active for a long time. So you only have to take one pill a day, usually only for about 5 days, I think. So it's kind of an easy one to kind of remember to take. You don't have to take it a bunch of times. Has some activity against both gram positives and gram negatives. Again, it's nice, you can take it orally and it's only once a day. There are some allergies to this as well, but again, if people are allergic to penicillin, then it's nice, they can take this drug. There are some GI issues with this. Another macrolide that I used to have to take was erythromycin, and that one, man, it would make your stomach cramp up, and I remember feeling like I couldn't eat, you know, the whole time that I was on it. So azithromycin, the GI issues aren't quite as bad, but sometimes still experience a little cramping like that. Now, if you remember fall 2020, azithromycin was one of the drugs that, you know, back at that time, we were just testing any drug that we knew was safe for humans to see if it worked against COVID. And for a while, azithromycin was on the news because, and you know, all kinds of alarm bells were going off in my head, like, wait, this is an antibiotic, this is not going to work against COVID. But it turns out that azithromycin has some kind of immune boosting activity. It kind of increases the production of some cytokines, which are messengers between our immune cells. And it also suppressed inflammation just a little bit. So we thought that, and it turned out that it didn't, in studies, they found that it didn't have any effect, but it might have remembered that from a couple of years ago. Okay. Our next class, we just have the nucleic acid inhibitors, and so there's just a couple of drugs there, and then the sulfa drugs, which block a metabolic pathway. So we'll finish up with that on Wednesday, and we'll probably get to resistance, most of resistance. So have a good day.